Hello everyone, welcome to Chemistry 251 Chromatography Lab. So we're doing thin layer chromatography, and chromatography is a technique we use for separating mixtures into their constituent parts. It relies on the different properties of compounds interacting with the stationary phase. So the most commonly used in chromatography is TLC, thin layer chromatography. It is basically a plate made of either plastic, in this case plastic, or glass, a thin piece of glass, and one side of the plate is coated with a thin layer of silicon dioxide, right? So using this technique, we are going to analyze how different compounds can separate according to their polarity. So we are given a set of standards: acetaminophen, aspirin, caffeine, salicylamide, and a mixture of all these four, reference mixture, along with the four unknowns. So these are unknown analgesic drugs, right? So it could be one of these four, right? So it could be anisine, Excedrin, BC tablet, or Tylenol. Okay, so these are four unknowns. We just don't know which one is which, right? And comparing this unknown TLC with the standards, we can then determine what type of analgesic and their components are present in each of the unknown. Okay, and TLC is going to tell us that. So I've prepared all the standards and the unknowns as well. So I took 0.2 gram of acetaminophen, right, and dissolve it in 50-50 dichloromethane ethanol mixture, right? So all these standards are prepared by weighing out about 0.2 gram each and dissolve it in 5 ml of solvent mixture. Okay. For the tablets, I took about half a tablet of each of the unknown, crush it, and dissolve it in 5 ml of 50-50 dichloromethane and ethanol. Okay, so these are all prepared, and all the drugs are well dissolved in the mixture. So we're ready to spot TLC plate. So before you spot the TLC plate with all the standards and unknown, you have to prepare the TLC plate first. Okay, first you draw a line 1 centimeter above the bottom of the plate, about a centimeter high, and you draw a line with a pencil, right? So here I've shown I drew a line using a ruler, draw it across, and I put a little ticky mark, little spot with a pencil. I mark it. That's where all the standards and the unknowns will be spotted. So I have them equally spaced. And next I'm going to label them first using a pencil. Make sure you lightly label the TLC plate with using pencil. Right? I'm going to label them before I spot the sample. Okay? So the first spot is going to be acetaminophen. So I write here, just write an AC. The second is aspirin, so ASP. The third one is caffeine, so I just put CF. And the fourth one is salicylamide, so SAL. And the fifth one is reference mixture, so REF as reference. And I'm going to pick one of the unknowns as well. So let's say I have unknown number 4, so I'm going to put 4, right? So this is how I pre-label the plates before I spot them. Okay, so I know exactly where I'm spotting and that I'm not overlapping the spots, and I have to make sure that everything is starting at the base equally at the line drawn across here. Okay? Okay. These are called capillary tubes. So thin glass pieces of tubes. We use this to draw out some sample and spot it on TLC plate. Okay, so these are very thin capillary tubes I have prepared earlier. Okay, so I'm gonna spot acetaminophen. So dip it, right? So you dip it with capillary tubes. So the small amount of volume solution goes into a capillary tube, and you're gonna spot it on the little ticky mark that I made here. Okay, and I blow it. You see the wet spot there at acetaminophen, yeah? And once it's dry, I'm gonna spot it again. Okay, and I'm gonna shake it dry because it's wet, right? I do not want the spots to be overly big. Okay, I want the spots to be really small, as small as it can be. So my question is, why do you need to make sure the spots are small? Okay, I'm gonna give 3 spots. Okay, that's good for acetaminophen. It's still kind of wet. Just gonna shake it dry. So I'm going to change the capillary tubes, use another one, a new one, to spot aspirin, right? So aspirin is in the second position. So I dip it, right? And then I'm going to spot it. I want to make sure the spot is as small as it can be. Right? So if you make the spots too big, then they're gonna overlap, right? Acetaminophen and caffeine and all the spots will be overlapping. You don't want that. So to prevent that, you spot it lightly and then dry it. And you spot it again. So the spots remain small in diameter, right? You want a small spot instead of a huge spot. Okay? You don't want the spots to be overlapping or touching each other. Okay? I spot aspirin a little more because it's difficult to see under UV light. So you notice I'm making sure that I'm using new capillary tubes for each standard, right? I do not want to cross-contaminate all the standards and then I can't figure out which compound is what, okay? So stay a little bit wet, so shake it dry, right? So I'm going to do this until the number 4, okay? You don't have to watch all these. So I have completed all the standards and unknown mixture spotted on TLC plate, right? And they are all dried, which is good. You don't want them wet, so all the spots should be dried as well. So next I'm going to prepare the TLC developing chamber. So the developing chamber is basically, this wide mouth glass with the lid. And I'm going to pour in ethyl acetate with 0.5% glacier acetic acid, right? So I have a big jar here. So I'm just going to pour in about 10 ml just to cover the bottom, right? You don't want to add too much because remember your TLC plate, all the spots are just 1 centimeter above the plate, right? So if the solvent is too high, then all the compounds will just flow backwards instead of capillary action rising to the top. You want that. And so if the solvent is too high, it will just redissolve and get dissolved into the solution, which is mobile phase, right? So this solvent is called the mobile phase. I'm just going to add a little more just to cover the TLC plate bottom. Okay, and I'm going to add in a piece of filter paper to saturate the chamber, as well as push it down a little bit. Saturate the chamber with the vapor, right? You want the ether acetate vapors. I'm just gonna stick it to the wall. Okay, it will also hold the TLC plate upright position. TLC plate is kind of flimsy and thin, so it's difficult to stand on in the chamber. Just add a little more, because yeah, that's good enough. So it's good, and you want to lower the plate into the TLC developing chamber, right? So I just hold it and slowly put it down. Okay, and close the lid, right? And from here, I want to make sure that it doesn't move, so you shouldn't move the chamber. Okay, you can see the capillary action rising, right? You see the mobile phase, which is the solvent, ether acetate with 0.5% glacial acetic acid. That is the mobile phase. And what is stationary phase in this case? Well, you can think about it, right? Okay, stationary phase is the one that's not moving. So what is this? What is that plate? Is that the TLC plate, silica on TLC plate, consider stationary phase? Well, I don't know, so you have to think about it. So I'm gonna let this TLC develop in the developing chamber, and we'll come back to this later, okay? So I think it is done. I'm going to pull it out, the TLC plate, so it is developed. You see it is about a centimeter above the TLC plate right here, just a centimeter below the top of the plate, right? That's where the stopping point is. I'm going to make a ticky mark before it completely dry off. So there, right? So this is the solvent front. That's where I stop it, right? Okay, and I'm gonna let this plate dry off. Okay, so I'm just gonna shake it and let it dry. And once it's dry, we're going to visualize the plate, visualize the spot using UV light, right? So we're not doing iodine stain. So we're going to use UV lamp and look at the spots, okay? So this is our in-house UV visualization chamber. So I just made a cardboard box, right? And you will see a UV lamp in there with the TLC plate that we just developed. Okay, so I'm going to turn off the light so you can see the UV lamp better. Okay. Okay, so this is the TLC plate under UV light. So it is under short wavelength, right? So double check. Yeah. So it's short wavelength, and this you can see all the spots that showed up on the TLC plate, right? So the TLC plate is coated with fluorescence material, right? So the plate is going to glow under the UV light, and the spots are going to show as a dark spot, right? Because they are not UV active, they are not fluorescing, so it shows as a dark patches, right? So that's how you can visualize the components and the standard mixture. Okay, so I'm going to circle them, right? So you're going to circle each spot. I'm going to put a little dot in the middle as well. right? So just to give the location where we have to measure the retention factor, right? You notice some of the spots are bluish, right? What are those? Are they fluorescing? Yeah. So I'm going to circle what I see, right? Okay, so this is using the short wavelength. Okay, I'm going to use different wavelength, longer wavelength, to see if there are any compounds that's going to fluoresce. Okay, so this is longer wavelength, right? So this is a longer wavelength. You will see spots on the top. They are bluish color. So I'm going to cross them, right? They are fluorescing under long wavelength. Okay, this one is just the bottom of the plate is just chipped, right? So don't consider this. Okay, so this is what it looks like on the TLC plate under both short wavelength and the long wavelength. Okay. So next we have to measure how far these compounds traveled up the TLC plate, right? So we need to calculate the RF value of each compound. RF value is the measurement of distance traveled by each compound divided by distance traveled by the solvent front, right? So I drew a line on the top here. That's the solvent front. So I have to measure those. All right, let's do that. So using the ruler here, I'm going to line them up and try to zoom in. Okay, so this is, this is about, so measuring the solvent front first, 5, 6, 7. So it's about 57 millimeter. So from the bottom here, the bottom line, to the solvent front is 57 millimeter, and from here bottom to the center of the spot, the first spot is acetaminophen, right? So I put a little dot here. So from here to here is measured around 26 millimeter. So I plug this number together here, and I have, let's zoom out. So into this formula, right? So 26 divided by 57, that will give me 0.46, right? So this is the RF value for acetaminophen. Okay. So calculate all these RF values for each compound, and you compare the RF values with compound unknown analgesic number 4, right? Here in this example, I'm using number 4. Okay, compare the RF values and also whether it's fluorescing under the long or short UV light. So you want to note those, whether I cross it out and under UV light, both long and short wavelength, right? So I took a picture of all these TLC plate, and I'm gonna post it on Canvas. Okay, so from the Canvas you will see all the pictures of both short wavelength, long wavelength, and the measurement for the RF values. Okay, you can do the calculation and the measurement. So I will post the pictures up there, and then from there you can determine the unknown analgesic, right? So the unknown can be from anisine, it can be from Tylenol or BC tablet, right? Or it can be Excedrin, right? So one of these your instructor is going to assign to you, so you can determine that from a TLC plate.